And I think at this point,
maybe another introduction.
An introduction, and so we've got this big front that we've opened. A way to start is to go all the way down to the infinitesimally small and start there. So we're very fortunate today to have with us Professor Martin Karplus, and I'm going to do a quick introduction, although you've so accomplished, this may take a while. Received his Bachelor of Arts from Harvard College in 1950, and worked on his PhD at Caltech in 1953. He worked at Oxford University as an NSF postdoctoral fellow from 1953 to 1955, when he joined the faculty of University of Illinois. In 1960, Dr. Karplus became professor at Columbia University in 1966 at Harvard University. He was named the Theodore William Richards Professor of Chemistry there in 1979, and he also became the Professor Conventant at the University, you can mock me later, Pascal, my pronunciations, University Louis Pasteur. He's a member of the National Academy of Sciences, the American Academy of Arts and Sciences, and a foreign member of the Netherlands Academy of Arts and Sciences and the Royal Society of London. He is a commander in the French Legion of Honor. You guys got a lot in common there. And has received honorary degrees from the University of Sherbrooke, the University of Zurich, and the Bar Ilam University, as well as numerous awards for his many contributions to science, including in 2013, the Nobel Prize in Chemistry. Over the years, Dr. Karplus has concluded and conducted research in many areas, theoretical chemistry and biochemistry, and has presented his results in over 800 journal articles and book chapters, as well as 2 books. His primary interest has been to develop and employ theoretical methods for increasing our understanding of chemical and biological problems, and his contribution has been instrumental in the transformation of theory from a specialized field to a central part of modern chemistry and, more recently, of structural biology. It is my great pleasure to welcome Professor Karplus to the stage.
Okay.
Thank you so much.
And thank you for being here, Professor.
It's a pleasure to be here. Can people hear me in the back? Well, modern technology is really great. Take me a second to open this machine. Oops. The lectern is made in such a way that it's almost impossible to get down to your computer, but we'll look at it. But while I'm getting this ready, let me make 2 comments. One is how wonderfully this meeting is organized, and if Dassault is as well organized as the way this meeting has been so far, I think they're doing a good job, at least in organization, which, from what we heard, is trying to bring together many different things to having them work together in an effective way. Now, the second comment I wanted to make is I was looking at some of the posters, and I saw what turned out to be a heart simulation. But to me, it looked like it was a knee simulation. And actually, I want to give simulia, these names are very hard to, you think simulia, as this, well, you can think of many things it might have to do with, none of which are simulations. But anyway, I have an artificial knee, and so far, artificial knee surgery is very far advanced, and if you have a knee replacement and do all the exercises that they tell you to do, you can get back completely this type of motion. But what they haven't been able to do so far is to give you the twisting motion. And what my surgeon says is, well, we have it to have it so tight, so you can't really twist your leg easily. And if you try to get into a car without twisting your leg, you won't get in. And so I would urge all these simulia people who design artificial hearts to make that their next problem. And And given, what we've heard and what I've saw, I really hope you can do this. I think this is a real problem, a real life problem, and I hope we can go forward. Now, when BioVia was introduced at the 3D experience in Paris, or in the neighborhood of Paris, I realized that there clearly were things that we had in common. It was, as Bernard, Charles, and Patrick mentioned to me, Dassault Systèmes had tried to use what they knew about airplanes to design proteins. Well, they're not that different, and they thought they could do all this, but they discovered that didn't work. And so what does a big company do? If it can't do something, it buys another company, BioVia, that is an expert in this area. And that's really how we got together. Now, I gave a talk at this introduction of BioVia in Paris with exactly the same title, and what you will hear today is an updated version of this talk. And so let me begin. This is a quote that many of you have seen, particularly if you're in the biological simulation area. Everything that living things do can be understood in terms of the jiggling and wiggling of atoms. Now, many people are more familiar, and this is something I want to read, with Richard Feynman, who won Nobel Prize in physics. But he was on the committee to discover what had gone wrong when the Challenger disaster happened in 1986. And what he discovered that it was really an unbelievably stupid mistake that had happened, costing the lives of these people. And so the committee didn't want him to say what he had actually discovered. But his comment was, and that's what I want to read, reality must take precedence over public relations, for nature cannot be fooled. I think this fits in very well with the ideas that we've heard from Bernard Charles, and perhaps to just remember to correct the pronunciation of one of my positions, but I have the title of Professeur Conventionné, which means you can do what you want in the University of Strasbourg, and you have no duties, but you can have students and everything. So let me now go on and look at the first protein. I mean, what I'm going to try and do, all of you know that living systems are made up of proteins, nucleic acids, lipids, fats, and sugars. And so most of the medications which are designed work on one of these that have gone wrong. To develop medications, it's useful to know how these systems function. And in my talk, I will concentrate on proteins because most of the medications that are in use are for correcting something that's gone wrong with proteins. And so first, I'll look at the function, and then in line with Feynman's quote, the role of motion in these functions. So let me show you first a video of Adelaide kinase, which basically takes two molecules of adenosine triphosphate, which is called the energy currency of the cell, and can convert them into one molecule. Sorry, let me say this again. It takes two molecules of adenosine biphosphate, PP, and converts it into adenosine triphosphate, this energy-rich molecule, and adenosine monophosphate. And this sort of potentiates the concentration of ATP, which we need in muscles and other systems, which I won't really be talking about. But let me show you an animation based on some of our calculations. This is an enzyme. An enzyme speeds up reactions, And what we see here, saw on the left slide, is two substrates coming in, the cap closing over these substrates, and then if we look at, so the reaction can take place isolated from solvent, and then of course, after the reaction, after one of the phosphates has been transferred, it has to open up and let the products escape. And if we now go back and look here at this structure, and one essential point is that proteins are developed by evolution, and particularly nature determines the structure such that the motions that are important for the function of this molecule require only a low energy. And in green, you can see in the slide hinges in this molecule. So essentially, the molecule has evolved such that these hinges are easy to bend around. And if you just bring in the two substrates, the energy of these two substrates is enough to close up the system. Let me now look at another, and maybe I should mention something that I was looking at, is what is the function of Adelaide kinase. It's involved in cystic fibrosis, and it's one of the systems that's essential for the channel function in lungs. And if it doesn't work, if there are mutations that make it inactive, is when you get cystic fibrosis. So in principle, one could try to work on this molecule to counteract the mutations, to use gene therapy to modify it, but so far this type of approach hasn't been used. So let me now go on and look at another molecule. It's kinesin, which is a molecule that goes from one part of the cell to another part of the cell, and it moves on microtubules. Cells, although if you just look at them, look like a mess, they're actually well organized, just like Dassault is well organized. And these microtubules are rails, essentially, which go from one part of the cell to another. And so at the top of this picture, which you can't see because one doesn't know the structure, are vesicles which transport the food materials or other things in a way that is very important for the cell. Now, just on the right is shown a slide. It's actually a photomicrograph enlarged about 10 million times, and you can see the kinesins walking on this. But let me now go back and look carefully at the kinesin walking on the microtubule. And if you look carefully, I won't go into the details, you can see it throws the left foot forward, and then the next step throws the right foot forward, and the next step again throws the left foot forward. If we wait a minute, it'll get there. There we go. So this system is really walking. Now, it doesn't walk like we do, but it walks like robots. And we've heard robots, and Simulia is interested in how robots walk, and just thought it was fun to show this slide because it walks really very much in the same way. Now, what's the importance of kinesin? I mean, one very great importance is that when cells divide, it pulls apart the two in a simple way, the two parts of the daughter cells. And so if it doesn't work, mitosis, which is the separation of the genetic information, is inhibited. I mean, that obviously can be a bad thing if you somehow have a disease of kinesin, but the contrary side of it is that kinesin inhibitors can be used for anti-cancer treatment. And there are actually a number of them that are being used, and essentially by making the kinesin inactive, it prevents the cells from dividing. Now this is, like all chemotherapy, you're depending on the fact that cancer cells divide 10 times or more faster than normal cells. So if you inactivate the kinesin, and if you can do this locally in the cancer, you're likely to kill the cancer cell before you kill the normal cells. Now another thing that it does, which is very important, is carry cargoes from the nerve body to the axons, which can be quite long. And if the kinesin doesn't work, you can't get the food from the cell body to the end of the nerve fiber or the dendrites. One curious thing is that viruses, they're very smart, and they have learned that if they jump on one of these kinesin molecules, they can get from one part of the cell to another in about 10 minutes instead of 10 hours that it would take them to do this by ordinary diffusion. Now, I mentioned the jiggling and wiggling from Feynman's lecture, and one important use of the jigglings, this low energy fluctuations just of the protein molecules, is that they can increase the range of possibility for inhibitor binding. And actually, recently, David Shaw and his collaborators at the Shaw Institute have shown something that's sort of a little bit surprising. You're always trying to find cavities in proteins that will bind a ligand and then inhibit the protein. And what they showed is that even though some of these fluctuations which open some hole that you don't see if you look at the X-ray structure are very rare, the increase in binding is sufficient to make this a actually interesting place for a drug to bind. Now the wiggling are these larger conformational changes, and which, like we've seen both in Adelaide kinase and in kinesin, and again drugs can be found to inhibit them. Now this just shows glycogen phosphorylase, which again is an important molecule because it's one way of attacking type 2 diabetes. It transforms glycogen to sugar. If you want to decrease the blood sugar, you can inhibit it. And it just shows a picture of all these various possible sites where inhibitors might bind. There's the allosteric site, which could be like in kinesin, the inhibitor site. There are a large number of sites, I won't go into them, and people have found a number of drugs which act in these various places. Now, one thing that we have been working on, well, for a long time, some of my students and I, is the question of covering chemical space. I mean, there are millions of different compounds that you can make, and actually now companies are storing these millions of compounds, and drug companies download them and try them on their target. But now, 20 or 30 years ago, we worked on virtual screening ideas and developed this multi-copy simulation search. And essentially, you take various small molecules that might be reasonable ligands, and you saturate the molecule with copies of these and find where one or another of them binds reasonably well, not very strongly. And here in particular, I mentioned there are 40 different fragments to look for these possible sites. And then by connecting these 40 different fragments in different ways, you can make a million drug candidates, and you pick out the ones, this is all virtual, which seem to bind best, and then you try to synthesize them rather than synthesizing a much larger number, or using the chemical library, which is relatively expensive. And this is just a very simple example of how starting with a compound that bound only with micromolar strengths by extending it using this type of method, you could get one which is a nanomolar binder. And sort of qualitatively, the statement is you really need nanomolar or sub-nanomolar binders to be interesting in terms of treatments. And I just looked up this morning how many references there were to this fragment-based ligand design, and there are over 1,000 of them. And so it's clearly a very important method. I don't know how much it's actually being pushed at Biovia, but it's something that some of the people here might remember. Now, the other thing that I talked about a little bit at the meeting was, I'm going to go over time a little bit, so don't count my introduction, which the slide here shows. It's the number of drug molecules that were made per $1 billion. And you see that in the 1950s, when this was all beginning and there were many possibilities, you got about 60 drug molecules for $1 billion. And as time went on, without going all the details, it became that nowadays you don't even get one drug molecule for $1 billion. And this is called Vroom's law, in the reverse of Moore's law about computing, which we could discuss here. But so there's a real problem. Now, what is the problem? In something that I've talked about, and certainly something that Biovia is one of the aspects that are very important, is that the drug design methods have improved tremendously. And up to the preclinical stage, we're doing much, much better. I mean, we're really, it's almost, if you say, give me a protein molecule that I want to inhibit, and give me a few months, I can find an inhibitor that we can synthesize. But then the problem is, if you give it to a person, it kills the person before it does what it is supposed to do. And that's where most of the drugs that are tried die. And so the question is there any hope of overcoming the failure rate? And that was actually the end of my talk in Paris, and I mentioned a number of different possibilities. And what I want to tell you about is what's happened in the intervening time. The one thing I suggested was increased understanding of the interaction of targets through system biology, which is one of the things that's being fostered now. And as far as I could find, there was no really direct evidence that so far was having an important effect on drug design. It has brought to the forefront the fact that most proteins that we have don't just have one function, but have multiple functions. And so you have to worry about that if you inhibit a certain protein, you may destroy other function that it has in the cell. And so I think in the future it'll become more and more important. Now, another idea was that increased focus on biologics, such as antibodies, would become more and more important versus new molecular entities, new compounds. And that's certainly happening. I won't go through the details of the slides. One of the very recent ideas, which hasn't so far worked actually, is to look at an entirely different kind of biologic, namely phages, when they try to enter a cell, lyse the cell so it's open. And so people are trying to use these lysines from the phages, which attack a cell from the outside in contrast to the inside that most antibiotics work. And there's hope that this will be useful. Now, I said new classes of diseases that can be treated with drugs. Of these, cancer and Alzheimer's are likely to be most important. There's no question that cancer treatments are really at the forefront of what's happening. Every year, there are maybe 20 or more different cancer drugs that are accepted by the FDA to be used. And I have some friends that have cancer, and as as the cancer has evolved in them, a new drug has always appeared when the old drug didn't work anymore. So this is, I think, a very hopeful area, and it says that 31% of the new agents that are used in the clinic come from cancer. And then finally, this was the idea that new methods of focused drug delivery based on nanotechnology could be useful. And again, there is real progress that functionalized nanotubules are being used for cancer drug delivery in mice. They haven't gotten to humans yet. And then there is this cute ideas, that's all I can call it, which is something that for BioVia is the sort of thing they might like to invent, but somebody else has already invented it. And this is to use the nanotubules as essentially a little cannon by putting in a fluid which evaporates, and when it evaporates, it shoots out the bullets, which, again, in the future idea, would be drugs. And these nanotubules, just as I said above about nice, can really be focalized in a very specific place. And something that I didn't mention at all, but is extremely important. You remember I said that it's easy to invent ligands that will do something to a protein, but most of them don't work because they kill you, essentially. And so people have now a number of ways of actually, rather than making clinical trials and all that, of testing both the toxicity and efficacy of various chemicals or biologics that they want to use. And given that time is up, I won't go through the details, but basically the idea is that you can mimic what happens in the body in the laboratory and see whether the chemicals are toxic. And mini brains have been made, sort of brain fibers in a row, and you can see whether the drugs actually work without going to human beings, which, as any drug company knows, is the largest cost of getting any drug on the market. Thank you very much. Oh. And it's been come up, at least in our conversations. Give me one more minute. I just discovered that I had some slides of my recent trip to China here, and I just thought I would show you a few of them. So, thank you very much again.
Thank you, Professor Karplus. You want to take us through some of those pictures from your trip?
Well, I mean,
we need to get you on Snapchat so that we can all subscribe, or Instagram.
I think just having shown them sort of a little bit on the side, perhaps.
You like to do the photography, right? That's one of the
right.
avocations.
Yeah. I mean, it's one of my friends, Jean-Pierre Changeux, who is actually a neurophysiologist, a very important one, has said that every person has his secret garden, and my secret garden is photography.
Wonderful. Thank you for sharing that with us.
So, should I just leave it?
I'll take care of it. Yeah. We're just gonna take a break, and then I'll come and help out. Well, thank you very much, Professor Karplus. That was wonderful. I had the good fortune of actually being in Vélizy, just on the outskirts of Paris, to actually hear the presentation, hear the previous material going back a little over a year ago. And Professor Karplus, I like the way the story is continuing. The journey continues, and as with science, it never ends, so we need to continue to prosecute that. Speaking of ends, we're at a break point in our material today, so we're going to take a approximately 30-minute break. We'll be back here in this room at 3 o'clock. Before you leave, though, really, really important, as you walk out the door, immediately to the left is the playground. We've got the posters and the partner exhibition area, and I really encourage you to get a chance to get out there and mix that up, check it out, and see what's in the pavilion. All right? Thank you, everybody.